Good morning. It's April 6th and this is your daily brief in science. Here's everything you need to know. A juvenile gray whale nicknamed Willy was found dead after a rare 20-mile upriver swim in the Willy River, a hunger-driven foraging move during the gray whale spring migration. The ascent occurred as gray whales head north along the Pacific coast, with many individuals entering Willy Bay on their journey from Baja California to Alaska. The whale was discovered near Raymond, Washington, as researchers monitor ongoing coastal migrations and track the broader movement of the population. NOAA Fisheries emphasizes that people should not interact with wild marine mammals and should observe from a distance to protect the animals' welfare. Cascade Research Collective scientists are monitoring the situation and urge the public to avoid close contact to prevent stress-related harm. Observers note broader conservation concerns for gray whales due to declines in Arctic prey and ongoing migration challenges. Cascadia Research Collective reports the whale appeared thin but had no visible injuries, and researchers plan to examine the carcass to determine causes and collect data. A necropsy is planned to determine exact causes and contribute data for ongoing studies, potentially as soon as the coming Monday, while giving the whale space. Authorities have previously declared an unusual mortality event for eastern gray whales spanning late 2018 to late 2023, with hundreds of strandings and a population around 13,000 in 2025, the lowest since the 1970s. Scientists attribute ongoing declines in eastern Pacific gray whale food availability tied to reduced Arctic prey to malnutrition and emaciation across the population. Experts indicate that fasting during the northward migration can push exhausted whales to seek new feeding grounds, possibly explaining inland river forays. Residents and social media captured the Willy River entry on the North Fork, about 185 miles southwest of Seattle, drawing crowds to bridges and viewing spots. A new vegan cheese aims to be healthier and greener, potentially reducing saturated fat to as low as 3% while improving meltability and an oozy texture beyond many current vegan options. Led by Professor Stephen Houston and funded by UKRY's EPSRC, the project seeks a heart-healthy cheese that matches market flavor and texture without sacrificing taste. The effort is supported by peer-reviewed work published in Food Chemistry, underscoring scientific rigor behind the development. Researchers emphasize that conventional vegan cheeses often carry high saturated fat, sometimes up to 25%, and rely on palm or coconut oils tied to environmental and wildlife concerns, including deforestation. The new process traps liquid oil in a 3D structure to mimic solid fat, reducing reliance on coconut and palm oils and aiming to improve health and sustainability. Professor Houston notes that traditional vegan cheese typically lacks protein and relies on starch and fats, with fats usually from coconut or palm oil contributing to higher saturated fat. Funding for the research comes from UKRIEPSRC, and findings are published in a peer-reviewed journal. A key goal is to use UK-grown crops to cut food miles and to test the cheese in a kitchen and with a tasting panel within about 10 months. Current lab results show potential to lower saturated fat and improve texture, with real-world tasting on the near horizon. The update highlights sustainability by prioritizing domestically grown crops and reducing deforestation risks associated with palm and coconut oils. A new angle on mental health places mitochondria at center stage, not just as powerhouses but as regulators of mood and cognition through energy flow, calcium balance, and immune signaling. Mitochondrial dysfunction is linked to major psychiatric disorders like depression, bipolar disorder, and schizophrenia, adding a complementary layer to the neurotransmitter story of dopamine and serotonin. In this view, mitochondria influence neural signaling by coordinating energy supply, calcium dynamics, and intercellular communication within the brain. Evidence shows cell-free mitochondrial DNA rises in cerebrospinal fluid and blood of people with depression, suggesting stressed neural tissue releases mitochondrial contents as distress signals. Extracellular mitochondrial DNA is elevated in depression, though the exact mechanisms are still being clarified. Overall, the presence of extracellular mitochondrial DNA in cerebrospinal fluid and blood points to a distress signaling role for mitochondrial contents in depression. Beyond cells, mitochondria can travel between cells and may act as distant messengers, expanding brain-body communication beyond traditional neurotransmitters. Extracellular mitochondria can circulate in blood and transfer between cells via astrocytes and platelets, signaling and potentially restoring function in damaged neurons. This extracellular mitochondrial communication represents a novel pathway for coordinated signaling between the brain and body. Mitochondria also modulate neuroinflammation by influencing microglial activation and cytokine production, which can reduce neurogenesis and synaptic connectivity and may affect how antidepressants work. Damaged mitochondria release DAMPs that trigger microglial cytokine responses, potentially lowering serotonin signaling and contributing to antidepressant resistance. Mitochondrial-driven neuroinflammation can impair neurogenesis and synapse formation, linking energy dysfunction to mood disorder pathology and treatment challenges. A 500-million-year-old fossil from Utah shifts the origin of chelicerates, the group that includes spiders, scorpions, and horseshoe crabs, by about 20 million years into the Middle Cambrian. Discovered in 1981 in the Wheeler Formation by Lloyd Gunther, and later analyzed by Rudy LaRossi Aubril, the specimen Megachelicerax cousteaui predates the oldest chelicerates from the early Ordovician Phezuata biota by roughly 20 million years. The fossil shows paired chelicerae beside the mouth, providing the earliest evidence of chelicerates and revealing a modern-style body layout for Cambrian arthropods. The genus name combines Greek roots for large, claw, and horn, while the species honors Jacques-Yves Cousteau, after whom the fossil is named. The specimen measures just over 8 centimeters and exhibits a 2-region body plan: a cephalic shield, 9 body segments, 6 feeding and sensing appendages, and book-gill-like respiration, plus the oldest known primitive chelicera. Its head shield carries 6 pairs of feeding and sensing appendages and dorsal plates that resemble modern horseshoe crabs, indicating advanced features in early Cambrian oceans. Megachelicerax cousteaui appears as a transitional form bridging Cambrian arthropods and later chelicerate relatives, suggesting earlier regionalization and chelicera development than previously thought. The discovery supports a Cambrian origin for chelicerates and implies that key body plan features were already forming shortly after the Cambrian explosion. Experts note a rapid Cambrian rate of evolutionary change, with early division into specialized body regions and the emergence of chelicerae reshaping our understanding of chelicerate origins. The chelicera distinguishes chelicerates from insects and is the pincer-like appendage central to their defining anatomy and, in some lineages, venom delivery. Overall, the study provides critical data for reconstructing chelicerate evolution, indicating that anatomically complex arthropods occupied Cambrian oceans and that biology and environment shaped their success. A new ultra-thin MOSE2-based grating could redefine how photonic circuits control light. Researchers grew large, uniform MOSE2 layers via molecular beam epitaxy, addressing previous exfoliation limits that limited films to tiny areas. The compact structure enables third harmonic generation, turning infrared photons into visible blue light, with the grating boosting efficiency by more than 1,500 times versus a flat MOSE2 layer. MOSE2's high refractive index slows light more than glass or silicon, allowing dramatic size reduction while preserving confinement. The sub-wavelength grating traps infrared light in a layer just 40 nanometers thick, over 1,000 times thinner than a human hair. This design confines and concentrates light, effectively acting like a near-perfect mirror and enabling manipulation at scales below the wavelength. The work, published in ACS Nano, is funded by European and Polish sources, including the National Science Center, the ERC, ADVANCED grant, number 101,53716, the Foundation for Polish Science, and the University of Warsaw. The advancement could streamline photonic technologies by enabling smaller, faster devices and scalable production for real-world use. It represents a pathway to more compact, efficient, photonic integrated circuits that could be produced at scale. The combination of large area growth, extreme thinness, and high optical confinement positions MOSE2 gratings as a notable approach for next-generation light control. This has been your daily brief in science. To read more about these stories, follow the links in the episode bio. You can also subscribe to these updates via email at www.brief.news. For more daily podcasts about the topics you love, visit brief.news/podcasts. Tune in tomorrow. We'll be back with everything you need to know.